organizers as well for the opportunity to share some of our data here. For me, it's the first time at this meeting, and I'm already looking forward to 2 exciting days, hopefully. So as Andrew already said, I come from a classical research lab, and you will hear a story from a classical research perspective. Okay, I have to do it like that. So, as also Andrew already said, we have an academic research part on PP2A and how this family of trimeric holoenzymes is actually regulated and achieves substrate specificity. On the other hand, we produce our own monoclonals. We offer this as an academic service facility, and we also choose from the literature targets which are of interest to the broader scientific community and generate antibodies and also out-license them actually to companies, like, for example, MIG-tag, T2A tag antibodies, and in fact, the world's first CRISPR-Cas9 monoclonal antibody comes from our lab. So the story I'm going to tell you today all is about the carboxy terminus of PP2A, of the catalytic subunit of PP2A, which is highly conserved from yeast to man and contains 3 potential modification sites. And I will focus actually mainly on tyrosine 307 phosphorylation and then also on the C-terminal leucine, which gets carboxymethyl esterified. Why is it interesting? Because more than 30 years ago in the 90s, an initial paper proposed that phosphorylation of tyrosine 307 should inactivate PP2A, which was quite interesting because some PP2A complexes have tumor suppressive activities. So in the cancer field, that steered some interest and was followed by nowadays already 250 publications. However, this modification was never shown by mass spec, but only with antibodies. And you might be shocked now because I explicitly named the companies, but most of these commercial antibodies were really badly validated. And I'm not going into detail here now, but over many years, we have tried to really investigate this modification and got conflicting data. So after a while, we got really fed up and said, okay, we have to do that carefully. And we really set out to do a proper validation of these antibodies, whether they really detect phosphotyrosine 307. So now I told you, we couldn't detect it by mass spec either under physiological conditions, but we could detect this modification when we overexpressed the CiteAb unit together with constitutively active SARC overexpression. So we thought we can take these conditions to actually scrutinize these antibodies. And we did that for wild type and a non-phosphomimetic mutant of the CiteAb unit, both MIG-tagged, in the absence or presence of SARC. So you can see here with the general phosphotyrosine antibody, huge upregulation of global tyrosine phosphorylation. This is our control antibody showing actually the levels of PP2A because it recognizes an epitope at the other end of the CiteAb unit at the N terminus. And then we tested more than those 4, but I show you these 4 because these are the most cited in the field. A rabbit monoclonal from Abcam, a mouse monoclonal Santa Cruz, and 2 polyclonals, one from R&D and the other one from Sigma. And I think you can immediately appreciate that Abcam and Santa Cruz show exactly the same pattern as our control antibody. So no hint whatsoever that they would be phosphospecific. The R&D showed an upregulation with SARC overexpression on the wild type, but it also cross-reacted with the phenylalanine mutant. And only actually the Sigma antibody gave the pattern you would expect from a phosphospecific antibody. Second control experiment, which everybody should do when using a phosphospecific antibody, is treatment with a phosphatase, calf intestine phosphatase. So same conditions essentially, mix C plus SARC, and compare the first and the last lane. Huge global tyrosine phosphorylation completely abrogated by treatment with CYP. And then Abcam, Santa Cruz, same pattern as the N-terminal antibody. R&D does show a decrease, but there is residual detection, clearly. And again, the Sigma antibody was actually fine. All the signal was gone. So already that told us, well, at least the Abcam and the Santa Cruz are not to target. They are not showing what they should show. However, in the literature, people have seen fluctuations with these antibodies. So we asked ourselves, where do they come from? And for time reasons, I can only show you one experiment. It's an ELISA on C-terminal PP2A peptides, unmodified, leucine 309 methylated, tyrosine 307 phosphorylated, or both modifications on the same peptide. And if you look at the first three here, I mean, it's clear no difference between unmodified and phosphorylated for these two. There is a twofold increase with the R&D, but all three antibodies are absolutely abolished in the recognition as soon as the C-terminus is actually methylated. And again, the sigma looking fine, a minor cross influence of the methylation, but I would also argue that if you have modifications so close together, it's really hard to get an antibody which is absolutely specific for the one side and not influenced by the others. But essentially, the whole model for 20 years in the field is wrong, because these antibodies do not show phosphorylation, but they actually show demethylation of the protein, and the phosphotyrosine-dependent inactivation of PP2A has to revisit it. So at that point, I just want to take a little step sideways to a serendipitous finding at the beginning of the project, actually. Here we overexpressed MYC-tagged PP2A, but we started with HA-tagged PP2A. And what we did find is that also the HA tag can get tyrosine phosphorylated. And then we looked actually at the sequence of the HA tag and the PP2AC subunit C-terminus, and there is some similarity. And the RNT antibody also detected, again by peptide ELISA, also phosphorylated HA tag. Well, that's bad for the PP2A niche field, I would say, but it might also be interesting for everybody in the room and for the wider audience that two HA tag antibodies are also strongly influenced by HA tag phosphorylation. The one giving a almost twofold increase, and the other one, the 12CA5, completely abrogated as soon as tyrosine 9 is phosphorylated. So even those seemingly unsuspicious sequences, which everybody puts onto his protein of interest for tagging, can have very undesired consequences when it comes to detection. But coming back to PP2A once again, so I've talked about the C-terminal methylation, which is actually a very important modification for the formation of some trimeric holoenzymes. For others, it's not. And this is in particular important because the tumor suppressive trimers are methylation sensitive. And where is the problem? Many companies offer general PP2A antibodies which were raised against an unmodified C-terminal peptide. So it could be that these antibodies actually preferentially detect this C subunit and not that one. And that's exactly what's the case. That was in the second study we published. Our trick is that we treat cell lysates with sodium hydroxide, which hydrolyzes the carboxy terminal methyl ester. So we end up with 100% non-methylated PP2A. And again, for time reason, I just show you three antibodies. All three show a massive preference for the non-methylated PP2A. And furthermore, PP2A having or sharing the same three amino acids at the C-terminus with two cousins, PP4 and PP6, they also cross-react with PP4. So these antibodies, which are sold by general PP2A antibodies, should actually not be used and are simply not suitable to investigate total PP2A activity and holoenzyme composition. And because this is such an important modification, we tried to solve this issue once and forever. So we generated our own monoclonal antibodies, which are super specific either for methylated or non-methylated PP2A, which do not cross-react with PP4 and PP6. And we have used the one, for example, in a collaborative publication as a marker, because carboxymethylated PP2A can really be used as a novel tumor marker for tumor progression in prostate cancer. And to round it up, and that was a huge effort for our small lab in the last few years, we have now a complete panel of total methyl and non-methyl PP2A, PP4, and PP6 antibodies, which for the first time will actually allow to really comprehensively study the methylation of all these PP2A family members in healthy and diseased cells. So that's the end of my data slides. And for the last 2 or 3 minutes, well in time actually, I think, let me talk about what was the consequence, because this was published right at the beginning of the pandemic, so quite a few years ago already. So what was really the consequence of us publishing this? It was well received in the PP2A field because that was an issue, and people now widely are happy with the fact that tyrosine 397 phosphorylation is not inactivating PP2A and might not even exist under physiological conditions. But if you look at then antibody validation and what the companies actually did, it's more of a mixed bag. And again, I'm outspoken with the companies. What has changed since 2020? Well, Abcam has discontinued their antibody in 2020. And I think that's what should happen. I mean, Abcam has hundreds of thousands of antibodies, and it would be nice if every antibody is super well validated, but we can see that this is very hard. And they just reacted the right way. Santa Cruz and R&D still offer their antibodies as phosphotyrosine 307 antibodies, and these 2 screenshots were taken on the weekend. Just to make sure that they didn't disappear over the weekend. And consequently, in 2021, since 2021, these antibodies have still been used by people as phosphotyrosine specific in more than 30 publications. So not by people in the PP2A field, but by people in the cancer field just touching on this issue. And it's really annoying, especially with Santa Cruz, I have to say, because I know, it's just another example. We contacted them, told them, please take it off the market. They said, no, what we are going to do is we put your paper there as reference, and then the customers can judge themselves. Well, they did that, but the reference list is this list. And then, well, our paper is somewhere in there. And I think you know from experience that you are not going to go through this list of publications, and you will never, ever find it. And the quality control, the validation data, I mean, I don't want to go into too much detail here because the left-hand side is incubated with the phosphospecific antibody. The right-hand side is actually a completely different plot incubated with a general antibody, which we actually also showed it's method sensitive, and then they just paste it together. But what's really astonishing for us even was the predecessor of this monoclonal antibody was a rabbit polyclonal that was before this infamous Santa Cruz story. Actually, you know the story probably. We had this polyclonal antibody in the lab. We bought it in 2013, and now look at the validation data in 2013 for the rabbit compared to the mouse monoclonal today. I think it hasn't changed very much. So the conclusions, and I mean, Andrew is sitting there and he has proposed this 10 years ago. You need to have proper validation, and we also need to put the pressure on the suppliers to follow certain rules. And if they don't, we must not buy it from them. It's our, I mean, in this case, it's our own fault. At the same time, we should also keep the pressure on our peers. When we get grants to review, when we get papers to review, it's a tedious work to go through the materials and methods, but we also should make sure that the antibodies they use are properly validated. On the other hand, we as researchers, trust is good, control is better. Do the validation yourself and teach your students to do the validation properly. Teach your students how to critically judge the literature, how to critically judge what's in the internet at the supplier's homepage. Then, I think yours was in 2015, Monia Baker's, like, blame it on the antibody was also this Nature comment in 2015. I think it's maybe not as bad anymore as it was, but there is still a lot of work to be done. And therefore, I'm very glad for meetings like this to keep it up. And I think this should be like kept for the future. But to end it on a positive note, the name giver of our institute, Max Perutz, once said, in science, truth always wins. And that might be immediately, but sometimes it might take a bit longer, but it will prevail. And with that, I just want to thank my boss, Egon Ogris, the first author, Ingrid Frohnam, our collaborators, and you for your attention. Thank you.